presentation about extremophiles on Earth and beyond. We have the pleasure of having today with us Ben Tatton and Professor Daniela Billi. So I'm going to introduce the speakers quickly, and then the floor will be theirs to talk about the wonders of extremophiles. So Ben Tatton is a PhD student at the Astrobiology Open University in the UK. He's studying the limits of microbial life and uses molecular techniques and modeling approaches to understand how microbes have evolved to thrive under some of the most extreme conditions on Earth. He aims to contribute to the understanding of how microbes may be able to survive elsewhere in the solar system and beyond. Professor Daniela Billi is leading the Laboratory of Astrobiology and Molecular Biology of Cyanobacteria at the University of Rome Tor Vergata. Her research aims to decipher the molecular basis of cyanobacterial adaptation to extreme conditions on Earth with a focus on hot and cold deserts, unravel the survival limits of desert cyanobacteria under non-Earth conditions by using astrobiology platforms outside the International Space Station and planetary laboratory simulations, and finally develop cyanobacteria-based technologies to use in situ resources available on Mars and the Moon to support human and space exploration. We are definitely very excited to receive you today. And for the schools with us today, just know that this will be a presentation of approximately 20 minutes. And after the presentation, you will be invited to ask questions using the Q&A panel or the chat, and we will send them the way of our speakers. So thank you again for being here, and I'll let you start the presentation.
Good morning, all. So I'll start off our journey through the solar system, discussions around extremophiles, and then I'll pass over to Daniela to continue on. So if you bear with me just one moment. Okay. Fantastic. I hope you can all see my screen well.
We actually can't right now.
Oh dear. Okay. How about now?
Go. Yes.
Super. Okie doke. Right then, so let's start. So our solar system and extremophiles. So when we're discussing extremophiles, the first thing most people think about, the first thing that most of us think about is these little guys, or if we're going to start discussing extremophiles, we can break down the word to get an initial understanding. So extremo comes from the Latin extrema, outermost, and phile, lover, or lover of the outermost conditions. And these little guys here, tardigrades, there's a very important distinction that we can make to begin with. So tardigrades, they are able to endure extreme conditions in a very specific state. They're fantastic little organisms, but what we're going to focus on today is something like this. So this is Pictophilus torridus, and is the world's champion at dealing with acidic conditions requiring it to grow. So we're going to be looking at organisms that require extreme conditions in order to survive, rather than just being able to tolerate them. So if we're going to talk about where life could possibly be, let's first understand how we define life and what life needs. So life needs essential elements. This is in the form of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, or what is colloquially referred to as chumps. So right here you can see a little bit of carbon on the left and sulfur on the right. It needs an energy source. So that energy source can be in the form of solar energy. It can be in the form of chemical energy, similar to what you might find at a hydrothermal vent. And it also needs water. And water is incredibly important because it's the universal solvent. And all the biochemical reactions that life needs to be able to function properly happen within the water. So this is our solar system, and based on what we understand about life, and that requirement for liquid water, we can start to define a region referred to as the Goldilocks Zone. So the Goldilocks Zone is a region where it's not too hot and it's not too cold. And liquid water can persist on the surface of the planet. So of course we've got Earth, which is roughly 70% water. There's lots of it. And then you've got Mars that's kind of just sitting on the edge, where in the deep past, we may have had liquid water on the surface. In fact, we're pretty certain we had liquid water on the surface. There's lots of evidence for that. But Daniela is going to speak to you more about that later. So as astrobiologists, we would love to be able to go to some of these outermost stretches of the solar system to start trying to find signs of life. But the problem is, many of these places are really quite far away. So 1.3 billion kilometers away. Or if you translate that to something that's kind of easier to understand, it would take us about 7 years to get to the outermost moons of Saturn, if we were to go look at Europa or Enceladus. So that's quite a long time for spaceflight. Okay, so instead, what we're going to do is we're going to go to places on Earth that, although they take a little while to go to, are a lot less than 7 years unless you are walking there. So some of these places as astrobiologists that we go to, in the top right, you can see some of my colleagues on Svalbard, which is in really high latitudes. It's cold all year round. We can start to try and understand how organisms that love cold environments survive. In the bottom left, you can see another one of my colleagues working in Ladakh in India, looking at really, really salty environments and how microbes or extremophiles are able to survive in these really salty conditions. And then in the bottom right, you've got another one of my colleagues, in fact, my boss, Professor Karen Alston-Francis, who is in Dalal in Ethiopia, who's looking at extremely hot, acidic conditions and how life might survive there. And all of these places, they represent somewhere else in the solar system, or help us to define a limit on where life could live. And that's what I'm interested in doing, along with many other people. So when we talk about analog environments, it can be hard to understand sort of how similar these places are to other places in the solar system, and the places we want to understand where life might be surviving. But one of these two pictures was taken on Mars in Jezero Crater by the Perseverance rover. You might have heard about it landing on Mars a couple of months ago in the news. And one of them is in the Atacama Desert. And if you look, it's incredibly hard to tell the difference between the two. I've played a little bit of a trick on you by putting them in grey, so you can't see the colour of the sky. But just from sort of the shape of the rocks and everything, so just between yourselves, if you have a quick think about which one looks more Martian to you, I'll pop it up now in 3, 2, 1. The one on the left is the Atacama Desert, and the one on the right is Jezero Crater in Mars. So again, you can see it's incredibly similar just from looking at it, the same shape of the rocks and everything. Yeah, it's a wonderful place. So I'm going to start our journey across the solar system. Now we've gone through that initial little bit, and we're going to focus on the second planet from the sun, Venus. So why are we interested in Venus? This is a picture of its surface. This was taken by the Venera probe that landed in the late 80s. It's incredible quality image. And as you can see, sort of the surface, it looks very barren. Maybe not somewhere you'd initially think to look for life. Well, if we're going to start talking about Venus, we need to understand that the conditions there are very, very hot and also very acidic. If we did manage to go there, it's a very hard place to survive. Now, pH is just a little bit of background information. On Earth, when we look at pH, it tends to range between 0 in some really acidic hot springs and 14 in what we refer to as soda lakes generally, or some hydrothermal vent systems. But if we were to go to Venus and have a look there, some of the clouds in Venus are actually at -11, which is crazy. That's crazily acidic. Definitely, it's hard to imagine anywhere on Earth that could possibly have such a low pH. So, as I mentioned earlier, you had this Goldilocks zone where conditions were just right to let life to survive. Well, we might be able to find sort of pockets of habitability, such as in the atmosphere of Venus, where the surface is far, far too hot, going up to several hundred degrees. And then you've got the Goldilocks zones at about 60 kilometers altitude, where the temperature cools sufficiently in order to have clouds condense in a similar way to what you find on Earth. But unfortunately, those clouds are at pH -11 and consist of sulfuric acid. So if you were to live there as a microbe, you'd need to have some very special adaptations that we're going to discuss in a moment. So if we want to go and understand what kind of adaptations could exist with these microbes, you need to have we need to go to places like this. So in the left-hand side, this is the lull. I mentioned it earlier. We go there to try and look at the microbes that are able to deal with very acidic conditions. Some of the pools there are pH 0, and incredibly high temperatures as well. And if you want to go look at really high temperatures and pressures to understand how microbes are dealing with that, we can go to places like the mid-Atlantid ridge, which is a picture here on the right, and that's a hydrothermal vent. So, as I mentioned earlier, extremophile, we can break down the name in order to understand what it's telling us. Thermo, coming from the Greek for heat, and phile, once again, lover. So you've got a lover of hot acids. And as I said earlier, the pH, which is controlled by protons and how many free protons are in a system. The protons in this diagram are these little orange blobs, and these type of microbes here, Pictophilus torridus, they have an ability to repel these protons with their outer membranes. So this bottom picture here, that's of Methanopyrus candeleri, and that's the world champion for being able to grow at high temperatures, as high as 121 degrees. So really, really, really hot conditions that under normal surface pressure, the water would actually boil. But because of the depth that some of these hydrothermal vents are at, it actually stops the water from boiling, and the microbes have adapted really, really strong membranes that are very tightly compact and compressed together by the pressure. That means that it's actually much harder as well for the protons to be able to go through. It keeps the cell membrane fairly solid. So these membranes are often very fluid. They allow things sort of through quite easily, like water. And these conditions that the microbes, the extremophiles, have adapted to be able to handle these conditions. So we're going to move on to a little bit further out in the solar system. We're now going to look at one of Saturn's moons that you might not expect to find life at, and is kind of a bit of a polar opposite to Venus. And this is Titan. And if you look at this image, you can sort of see something glinting off the surface. If you've ever been in an aeroplane and you've been flying over the sea, sometimes you might see the light catch the water, and you would be forgiven for thinking this was a lake. It'd be very easy to think this is a lake. If you look here, it's got all the signs of a lake. You can see what look like little rivers running into these lakes, and these images were taken by some fantastic missions that were coordinated by the European Space Agency and NASA. but these lakes aren't actually water. They're condensed methane. So it's so cold there that methane turns into a liquid. Now, could this liquid replace water for biochemical reactions to take place? I'm not sure. Is there enough energy in the system because it's so cold? I'm not sure, but these are questions we all want to answer. These are questions that, as astrobiologists, we're extremely interested in. So with that, I'm going to pass over to Daniela. Daniela is going to, she's going to take you for a journey along the rest of the solar system, discussing Mars and some other icy moons that maybe there's a little bit more of a chance that we can find life at. But Daniela, whenever you're ready, you let me know. And
thanks, Ben. And no hello to everyone. I'm now sharing my screen. I think that you see it. Okay, I will put it in a full size. Okay, so good morning, everyone. And
Daniela,
as Ben,
I'm sorry, we don't see your screen actually. Actually, not on my side. So
I will do it again. 1 second.
Here we go.
Okay.
Perfect.
Okay, so as Ben was saying just before, it is really difficult to say if you look at a desert on Earth, if you are really on Earth or on Mars. In fact, there are places on Earth that they look really like Mars, as the Dry Valley in Antarctica or the Atacama Desert in Chile. And if you look at this place, you think that life is not there, so that they are lifeless as the surface of Mars. But thanks to expedition of several researchers, life has been discovered in this place. And we now know that microorganisms take refuge inside the rock so that they can have less extreme condition. And here you see a selection of rocks that has been colonized by microbes. The green layer that you see are indeed cyanobacteria that you see here. And there are these little guys, they are bacteria that are able to perform photosynthesis because they have chlorophyll, and they take refuge inside the rock or in the space between the rock and the floor. So if you just take a rock and you look underneath and beneath it, you can see this green layer that is made of cyanobacteria and other bacteria that manage to live in these extreme places that we really consider the closest place on Earth to Mars. Among these deserts, as I said before, the dry valleys are really very much interesting because life was discovered there in 1976. That was the year when the Viking rover that NASA sent to Mars failed to give us proof of life in the soil of Mars. So as you may know, we are really very much interested in searching life on Mars. But why? Because we know that at the very beginning of the history of this planet, Earth and Mars were really very similar. And there was liquid water on the surface of Mars. And as Ben said, when we look for life, we really have to follow the liquid water. But then, due to the small size of this planet, due to the absence of a magnetic field that is shielding this planet against the cosmic radiation and solar radiation, the atmosphere was reduced in size, and no liquid water could stand on its surface anymore. And Mars turned into a very dry and cold desert. So we know that in the surface there is no life, and this is also what the Viking mission at the very end showed to us. Because as I said before, in 1976, this rover collected some sample of soils and made some analysis, but it could not find a clear sign of life, in particularly when an instrumentation was used to look for carbon was not detected. And life as we know it is based on chunks, as Ben showed to you. And in a principal way, carbon is the most important element, and life is, as we know it, is based on carbon. So DNA, amino acid, all the building blocks of cell are made of carbon. But we now know why this failure happened, because in 2008, another rover, the Phoenix, discovered the presence of very toxic salt on the Martian surface, the so-called perchlorates. And when you look for carbon-based molecules using high temperature, as was the instrumentation on board the Viking, you just burn up the molecules. So this is the reason why, even if organics were present on the surface of Mars, these were not detected. And of course, when we talk of this, we always have to make clear that even if you find the organics molecules, you have to prove that are of biological origin and not of chemical origin. Nowadays, just while we are talking, there is another rover on Mars, the Perseverance, that is again looking for evidence or life on these planets, and it, of course, as a consequence of what we now know, is collecting sample not on the very surface, but 5 centimeters beneath, because we know that the surface is really not suitable for life. So in order to search for biosignature, that means for sign of life, we really have to look beneath it for the perchlorates, and also for, as I said before, for the high doses of UV and the ionizing radiation that are hitting the Martian surface. And the sample that are now being collected will be also sent back to Earth for further investigation, because the problem here is that if you want to use some specific instrumentation of Mars, you have to miniaturize it. So this is not possible with all the instrumentation that we have in the laboratories. So it will be very important to have this sample back for analysis. And next year, an European rover will leave the Earth and will land on Mars. The name is ExoMars, or Rosalind Franklin, in honor of the researcher who contributed to the discovery of DNA. And this will be very important because this rover has a drill who will penetrate about 2 meters into the Martian soil and will collect and will analyze the samples in situ. And 2 meters is important because this is a point, and it is a region under the Martian surface where there is no perchlorate, so there aren't this very toxic salt, and also there is some kind of protection of shielding against the radiation that are hitting the Martian surface. On Earth, there are other places very important as an astrobiology target for searching life, that are the subglacial lakes in Antarctica that really look like 2 icy moons of our solar system, Encelado and Europa. As Ben was showing you, these 2 moons are far from what is the habitable zone of the solar system, so are in a place where liquid water can't be present on the surface, but we know that they have liquid water under the icy crust. And so if liquid water is there, this is really a key ingredient for life. And on Earth, we have a very similar place to this, that are the subglacial lakes in Antarctica, like, for instance, the Lake Vida. And what the researchers did is that they drill the icy cover of this lake, and they reached the place where liquid water was present at very low temperatures, minus 10 degrees, due to the presence of high concentration of salt. And life was there. There were microorganisms, bacteria, that were living in the dark, and also in the absence of oxygen. And the point of oxygen and dark is also very important to search for life, because, as Ben was saying, the cell life needs an energy source. That could be the sun, but could be also another kind of chemical source of energy that is present in the dark. So this kind of life in the dark and in absence of oxygen is really very important for searching life beyond Earth. And so these subglacial lakes are very similar to the two icy moons, Enceladus and Europa. And these are important not only for the presence of liquid waters, that is only one prerequisite. They have other ingredients that are important for life. First of all, organics. As I said before, life as we know it is based on carbon, and the building blocks of a cell are made of DNA, amino acids, so molecules that are built using this element. So this is very important. And these organics, as we detected in the plumes, that are a sort of cold geyser that are ejected outside this icy moon. And this has been studied by the Cassini mission. And the Cassini saw that the hypothesis that the organics that has been detected in the plumes originated in the core of this icy moon, and then they found their way outside. So there is a place of liquid water where life may exist, and also in some places of this icy crust. And the other important discovery is the presence in the plume of methane, because the presence of this gas, this time is the liquid, is not it's a gas, not a liquid, as Ben was saying, talking about Tatton, is important because it is associated with some reaction that has been associated with the origin of life on Earth. So let me explain this a little better to you. On Earth, we think that life might have originated in the hydrothermal vents, like Ben was showing you before, and these are a sort of hot geyser in the floor of the ocean, where actually the water of the ocean enter the bottom of the ocean and then get heated by some kind of reaction that we think may happen also in the icy moon, because they are associated with the release of methane. And while getting out from the bottom of the ocean, they form this kind of structure. And this is what we call hydrothermal vents. In our days, these are colonized by thermophilic microbes. But we think that the origin of life on Earth happened here. And this was the place where from simple elements, reaction occurred, so that we moved from a world of molecules to a world of cells that then left this environment and colonized the Earth's surface. So we think, based on the evidence that we have, that this might have happened also in Enceladus, but the same applies also to Europa, the other icy moon that is very relevant for astrobiology. So as we said, we really have an idea of what life can face on Earth by looking at extreme environments and looking and studying extremophiles, but we can also extend our knowledge on the limits of life by exposing these extremophiles to non-Earth conditions, for instance, by using a scientific box that is installed outside the International Space Station. But we are interested in searching for life not only in our solar system, but also in other planetary systems that are orbiting other stars. So here you see these blue lanes, that is the habitable zone. So in our system, as Ben was showing to you before, the Earth is just right. The Earth is just at the perfect distance from the sun, so that liquid water can be present on its surface. Mars is just outside this very special place. And if we look to other planets, we can say which one is potentially habitable, because it is at a certain distance from its star, so that liquid water can be present. And when we search for signature of life on exoplanets, we can look at the presence in their atmospheres of gas that we associate with life, like oxygen. And we look at this by using space telescope or even Earth telescope. And the oxygen, as we know, is really linked to life on Earth, at least linked, as I said before, to life form that utilize the sun, like cyanobacteria, algae, or plants, just following the evolution. And we know that these, for instance, cyanobacteria can produce, are able to photosynthesize using visible light. So when you study photosynthesis, you talk about using visible light between 400 and 700 nanometer in the wavelengths of the light that is emitted by our star. But we now know that cyanobacteria, when exposed to a wavelength longer than 700, so in the far red, they can produce a new kind of chlorophyll that are able to absorb and so allow the photosynthesis behind the visible zone. And this is really fantastic, because we know that many exoplanets that are orbiting M stars received a spectrum that is very different from that of the sun. Here you see the spectrum of our star, and here you see the spectrum of M star, and you see that it's really shift towards the red, the infrared. So based on what we know some cyanobacteria can do under simulated conditions on Earth, we can speculate that actually photosynthesis can happen on this exoplanet. So thinking about other potential habitable and maybe inhabited planets, we thank you for your attention. Many greetings from me and Benz.
Thank you very much. Thank you very much, Daniela. Thank you very much,
welcome.
Ben. This was absolutely exciting and fascinating to hear about this journey across our solar system and how we can push the limits of life. This was very interesting, to say the least. We already have plenty of questions for you in the chat, so let me go through those. The students of Marina Mola in Greece are asking a lot of questions, so I'll take them in order for now. The first one comes from Vasilis. He's asking, why do we have extreme temperature conditions on the Earth's poles, actually? And this probably relates to the lake in Antarctica that you were talking about and these ice layers. So if one of you wants to take that question.
I can take that one.
Sure.
Kalimera, Vasilis. We have cold conditions on the poles because of the way the way the Earth orbits the sun and the angle of the Earth. And as we go to these higher latitudes, sort of the way that we're getting sort of temperature distribution around the Earth. Because of the angle that it's at, thank you, the way that we're getting temperature distribution around the Earth means that these regions don't get the same kind of energy from the sun that we have, say, near the equator, and also it's further away in terms of sort of like the sphere as well. So yeah. That's why we get these colder conditions.
Okay, thanks. Let's keep with a bit of scientific questions first. Vilda is asking us, what is the highest temperature Venus can reach? And this is coming from the KTU Lyceum of Engineering students.
I think that it can go up to several hundred degrees. I'm not sure the exact temperature, but I believe it's over 400 if my memory serves. I'm not too much of an expert on Venus. I specialize on Mars, kind of like Daniela. I think it's around the 400 degree marks at the surface. But like I was saying, the temperature on Venus really fluctuates as you start to go up through the atmosphere. So as you get higher, it goes as cold as sort of like -100 at sort of the edge of space for Venus, sort of goes all the way through a gradient, similarly to
to Earth. Again, if you go higher up in Earth's atmosphere, it gets much colder. That's why if you ever go on a plane and you have a look out your window, even though you can be flying over the equator, you see tiny ice crystals on your window where the air condenses and freezes because you're much, much higher, so the temperature's much lower.
Thanks. Maybe this one is for you, Daniela. So Vilda is also asking us, what about life in the sun? Why didn't we explore the sun earlier? And regarding the limits of life, maybe you can tell us about why we did not explore the sun for life.
Our sun, our star, you mean?
Yes.
Because this is, I think, like hell for every kind of life that we can think about, because just radiation will be, UV radiation, ionizing radiation will be really impossible for life, and there is no liquid water, and I think there are no elements, all the elements that life needs. So really, when we search for life, we don't search life on the star, but only on the planet orbiting the stars. And also, if these planets are too close to the stars to have liquid water on the surface, so that they maybe not even orbiting the star, but being locked. And then the problem is that the energy and the radiation that the star is emitting. So we really cannot think about life on a star.
Okay. I mean, this is an actual valid question. Like, just why not start with the star and then extend to the rest of the solar system? So thanks for this answer. Okay. Panagiotis from Marina's school is asking, is the aim of all these missions to find life, or are there other aims as well?
Well, may I take this? Usually
Sure.
Space mission has different target, and maybe the search for life is only one of these tasks. For instance, if we think about Perseverance, that is the second missions looking for life after the Vikings, the search for life is only one task. The other one is the study of the planet, but there is also one experiment that is studying the possibility to produce oxygen using the Martian environment. So this is in support of future human space, human outpost on Mars. So the production of oxygen using in situ resources.
Mm-hmm. Yeah, thank you. Okay, now let's move to more questions about your day-to-day life as researchers, where you come from as a researcher, what is your career like? So we have plenty of questions about that. Again, from Marina's school, we have a question. Were you a good student at school, and what science subject was your favorite one? So maybe both of you can actually answer this.
Do you want to go first, Daniela, and then I'll come in afterwards?
Yeah, I can. Well, I was good at school, nothing terrible, but I had a good attitude. But I really have to say that when I was studying also at the university, astrobiology didn't exist. There was only exobiology that was funded by NASA to search for life only on Mars. And then after the failure that I said of the Viking mission, there was a reduction in the interest for this kind of research. But then when actually the first exoplanet was discovered and we got the evidence of liquid water beneath Europa, and also the discovery of the Martian meteorites, but I'm not going to talk about this. There was a new interest in this, and so I have to say that the term astrobiology was coined when I was actually making the defension of my PhD thesis, and now I'm working on astrobiology that actually didn't even exist when I was a student. So I've been very lucky, and I wish to you the same, that by chance and by fortune you then end up in doing something extremely new for you that you like very much. Ben, to you.
Thank you. Yeah, so I was okay. I was an okay student. I think I was probably generally sort of like pretty middle of the class. Wasn't I wouldn't say I was one of the top students, but I'm dyslexic, so I found school a little bit harder with the reading and the writing. So I was more interested in football and music and my friends and things like this. And then it wasn't until I got a little bit older, about 23, because I'm nearly 30 now. So when I was 23 or so, I went to university, and like Daniela said, had a good attitude, but again, wasn't necessarily always top of the class, but I worked very hard, and was told about astrobiology by one of my teachers or lecturers, and I thought, wow, that sounds amazing, as if people are able to investigate what life might be like on other planets. That sounds like a really cool job. So after finding that out, I really focused hard, worked really hard, took what opportunities, was a little bit, took a few chances, took a few risks in applying for things that maybe I didn't think I would get. And yeah, here I am, currently in the second year of my PhD. So yeah, there should be nothing standing in your guys' way if you really want to go and do it.
That's fantastic, both of you. Very fascinating experiences. Marina is saying in the chat, wow, my students can relate, and thank you for sharing about dyslexia.
Yeah, no problem.
Very important. And she's saying we have students for the special needs class joining us. So yeah, well, thank you for sharing this.
Yeah, no problem. I was in the special needs class as well at points. So yeah, you guys, there's nothing standing in your way.
Thank you. And yeah, Daniela, it's super interesting to have your view about also the fact that you had an entire field getting created as you were going through your PhD and defending your PhD. So yeah, it's actually things that sometimes we don't see that much, actually, the creation of a field. So we think that science is just like those disciplines, that they exist and that's all. Then some get created, and then new jobs and new interests. So that's great. So yeah, going back to all this, I think you already touched on that, but Panagiotis was asking, were you always interested in astrobiology and science? So if you want to add anything about that.
I can just say one more thing that I used to remember, that long time ago, I was in Tallahassee, Florida, with Imre Friedman and his wife, Rosalie Ocampo, who actually are the two researchers who discovered life in Antarctica. And so I was there, and at that time, Friedman has also small pieces of the Martian meteorites in his laboratory. And I was there, I was really very young, and I really had no idea how this research topic was going to change my life. So it is maybe something that you then, when you think of the past, you realize that things happen that you really had no idea, and then they are going to change your life very deeply.
Yeah, I would say as well, I was always very interested in science. So it wasn't necessarily that I was really, really good at it, like the best in the class. But I think having an interest, sort of like a real curiosity for these things, really helps. It means that when maybe you don't want to do the work, or you're feeling a little bit fed up with your homework or things like this, having that curiosity really helps and makes you want to know. So yeah, I've always enjoyed science, but yeah, didn't always want to be an astrobiologist. Was going to try and be a marine biologist in Greece for a little while.
Thanks for sharing. A very tough question. I find it very tough every time, from Marina student Agelos, who's asking, what is your favorite discovery? Who wants to go first?
I don't mind, Daniela. It's me or you. I'm happy.
For me, my favorite discovery is the very last topic that I presented, that is the possibility of photosynthesis out the visible region. This is something that really fascinated me, because at school we just study photosynthesis and chlorophyll, chlorophyll A, chlorophyll B, and that life has to stay there. But the demonstration that on Earth, in some shadow places, we have cyanobacteria that can produce other chlorophyll and do, as I said, the photosynthesis outside the visible region, I think it's really one of the most exciting discovery from my point of view, because it's really something that you don't expect. And the message is that life is always doing something that you don't expect.
Wow.
Yeah, I have to admit, I think my favorite discovery, when Daniela told me about that, because I didn't know before Daniela told me, when we were discussing this, I was like, wow, that's amazing. So you're always learning from one another. So yeah, that's a really cool discovery. I think my all-time favorite discovery has got to be DNA being a double helix. It's beautiful. Yeah, it's one of the researchers that found it, along with Rosalind Franklin. Afterwards, they were from England. Famously, they went to the pub afterwards and said, it's so beautiful, it must be right. And I think that's kind of true. It just looks perfect. So yeah, I like the discovery of DNA and how it reproduces itself and all this kind of stuff. That's fantastic.
Very inspiring, yes. So the next question we have is regarding exoplanets. One of Marina's students, Agelas, is saying exoplanets are amazing. How can scientists detect them?
You know, the problem with exoplanet is that we cannot send a rover there like we do on for Mars, or that we will do for the icy moon Enceladus and Europa. So we can only look from Earth using telescope. So the only way is to look for signature of life in the atmosphere, as I said, or we can also look at signal coming from the surface. So it's like when you look at Earth, you have the green color that is due to the presence of plants and to the fact that chlorophyll do not absorb the red region of the spectrum and reflect, so you see green. So we can imagine to see planet if life is there and is using the light of the star that is shift. We can think about different pigments and different colors, so we can think about exoplanet of different colors, but we have to look from remote. We cannot go there. So even if, and we look for habitability only on the surface. So if there is places like our icy moon with liquid water under the surface and life, we will never know. So the only problem with that is that we look only for life on the surface that we can detect from remote, but we will never know about the subsurface life.
Yeah, it's there's lots of my colleagues that look at exoplanets as well at the Open University, and as the exoplanet goes past its star, so as it's going around it, you see a little dip in the light, and from that dip you can infer a lot of the information that people like Daniela use to be able to sort of unpick what life might look like there, and all those kind of things. But as Daniela says, if we had an icy crust and water beneath, we'd never know. It could be in the solar system and we don't know because it's below an icy crust. Who knows?
Yeah, this is actually one of those existential questions where you're like, oh, it's probably there and we will never know if we don't have the chance to just stumble upon it or something like that. Yeah.
Yeah, and the point is that we look for life, as we always say, as we know on Earth. That is our big limitation, because we have only one case of study, that is life on Earth, that is based on carbon. And so it is very, but as Ben was saying, the discovery of DNA is also basic, because life, as we know, it is based on DNA. And so if we have to give a definition of life, it is a chemical system that is able to sustain itself and to undergo Darwinian evolution. So it could be not really a cell, but just a molecule like DNA that is able to replicate and introduce mistake, and so adapt to an environment that is changing. So the definition of life itself, it's quite problematic, what we are really searching for.
Which is why it's important to always push the limits of life also, as you were mentioning, because this is how we can expand our imagination of what life might look like.
Yeah, for this experiment that I was mentioning before outside the International Space Agency, the International Space Station, or on the ground using facilities that are simulating high doses of radiation, that are simulating the condition of the icy moon, that are simulation M stars, and so see. So we really have to push the limit of life as we know it, taking advantage of space that is more extreme. Of course, the environment in space on the moon are more extreme than the environments that we have on Earth for certain point of view, and also make simulation on the ground in our laboratory, changing the pressure, the composition of the atmosphere, a combination of those, and see how much life can do. And I'm sure that life is always going to surprise us, because in every place we go, as Ben was showing, life is there. Even if you don't see it, maybe it is hidden, but it's there.
So, yes, Ben, sorry.
I was going to say the question, were you about to mention the question from Nasia?
Yes, I was going to mention that one, but I wanted to stay a bit on this topic first, because we have another question from Thanasis, who's asking, could a form of life be based on silicon? And I guess this is something that has been discussed a lot for a while. So what's your point on that, both of you?
The answer is that, of course, silicon can do the same chemistry of carbon. But the point is that if we look at the interstellar medium, molecules that are based on carbon are much more abundant than those based on silicon. So just based on this, life should be based on carbon. It is like the hydrogen, sulfur, phosphorus, nitrogen, hydrogen, and oxygen, that are the most abundant element in the universe, that are also the element that life used for the building blocks. So it is just a case of abundance. I don't know if Ben wants to add something else on this.
I would say that there's lots of silica on Earth, and we have perfect conditions for life to evolve on Earth. Maybe there are some conditions under which silica is more preferential than carbon, but I don't think so. None that I know of or I've ever read about. So I think if life was going to evolve based on silica-based or silica-based life was going to evolve anywhere, Earth is as good a place as any for it to have happened, and it didn't. So my instinct is, if we find life elsewhere, it'll also be based on carbon, because of some of the things you can do with carbon and how it's just such an interesting chemistry element to be able to sort of perform sort of what we understand as biological functions. So yeah, I think it's more likely to be carbon.
All right, thanks. So we have one last question coming from Marina's students. First, she wanted to share that she wanted to thank you for inspiring them. So the last question is, what subjects do you have to be good at to study astrobiology? And the question is coming from Nasia. Ben, do you want to go?
Yeah, sure. You can be good at any subject and do astrobiology. I work with some amazing people who study all aspects of astrobiology, and the astrobiology OU is a very inclusive group. So we have people who study the really hard science, who are doing the physics. Like one of my colleagues, she is studying sort of space instrumentation. She's an astrophysicist. Her name's Grace Richards. She's helping to design instrumentation that might be able to detect signs of life on Enceladus. So really hard science. But then we have people who I also work with, Dr. Alessandra Moreno, who is looking at the ethics around astrobiology, and another PhD researcher, Devyani Gaja, who is focusing on how space technologies can be used to help developing nations, and how you sort of have a disparity between sort of richer countries and poorer countries, and who actually has access to space instrumentation. And then there's people who work in law, who are looking at the law of sort of space, and that comes under astrobiology as well, because humans in space, it's biology in space. So yeah, you can study anything can be involved in astrobiology. It's just whether or not that's the path that you want to go down. You can go down the sciencey route, or more of the other humanities type routes, or social science. So you can do anything.
Yeah, indeed.
Excellent.
You can be a biologist, you can be a chemistry, if you want to look to the prebiotic chemistry and how molecules and how all started. You can be a geologist looking for biosignatures. So really, you can get to astrobiology following what you like most.
Thank you very much, both. Thank you very much to all the students who asked all those fantastic questions. I think, given the time now, it will be time to end the session. So yes, thank you again, everyone. It's been a pleasure listening to your presentations and being part of this lively conversation. Yes, any last words from from you before we end the session today?
No, just thank you for listening to both myself and Daniela. It's been a pleasure.
Yeah, it has been for me a very great big pleasure, and thank you both for the invitation, and Ben for sharing the floor with me. Amazing.
Thank you very much, everyone, and bye-bye. Have a good rest of your day.
Bye. See ya.
Thank you. Bye-bye. You take care.